A mutation is any random change in the DNA of a cell or an organism. DNA is a long, helical molecule with all the instructions for how living things will develop. These changes can happen either through external factors like radioactivity or UV and X-rays, or through internal factors, such as when a cell is dividing and it incorrectly copies the DNA.
An important source of genetic variation comes from mutations. A mutation is a change in the DNA sequence of a gene. Though some mutations are harmful, most are not. A mutation may change a trait in a way that may even be helpful, such as enabling an organism to better adapt to its environment. The simplest mutation is a point mutation. This occurs when one nucleotide base is substituted for another in a DNA sequence. The change can cause the wrong amino acid to be produced. In some cases, the change has little effect. In other cases, the incorrect amino acid can affect the structure or function of the protein being encoded. Here's an example of how this works. This sentence, composed of 3-letter words, is similar to a DNA sequence made up of 3 base sequences called codons. Now, what happens if one letter is changed? The sentence now has a different meaning. Notice that the change affects just one word. If this was a DNA sequence, a change in one codon might not cause problems, but in some cases, it can have a severe effect. For example, here's part of the DNA sequence for hemoglobin, a blood protein that carries oxygen to the tissues. Each DNA codon codes for a specific amino acid. For example, the codon GAG codes for the amino acid glutamic acid. The amino acids, in turn, are the building blocks for the hemoglobin protein. Now look at what happens when a point mutation occurs. This single mutation causes the amino acid valine to be encoded rather than glutamic acid. This one change has a critical effect on the structure of the hemoglobin molecule, causing the condition known as sickle cell anemia. Another type of mutation is a frame shift mutation. This occurs when a base is added to or deleted from a DNA sequence. Let's look at our sentence again. Now let's add a letter. This changes the message starting at the point where the letter was added. It affects that word as well as all of the words downstream, that is, the words that follow. The same thing can happen in a gene. Here's a normal sequence. Now watch what happens when you add a base. The downstream message is completely different. A similar problem occurs when something is deleted. Frame shift mutations can cause drastic problems. As you can see, none of the downstream amino acids in either example make sense for the protein they are encoding. Because there are so many changes to the amino acid sequence, the resulting protein most likely will not be able to function.
Chromosomal mutation. Chromosomes are the condensed and compact version of DNA, and chromosomal mutation happens when a section of a chromosome gets rearranged. Basically, a chromosome can be duplicated, inverted, or exchanged with another portion of the DNA, or it can simply be deleted. Scientists identified these mutations by bombarding fruit flies with X-rays and UV rays. These poor flies did not become superflies. They usually became very unsuper and abnormal flies. These heroic flies also helped scientists find another type of chromosomal mutation, called aneuploidy, where the total number of chromosomes could actually change. In most multicellular life, chromosomes come in pairs, one from the mother and one from the father. Humans have 23 pairs, making a total of 46 chromosomes, whereas a fly has only 4 pairs of chromosomes. Sometimes, when sex cells, the egg and the sperm, divide, the wrong number of chromosomes end up in the cells. In the resulting cells, one might have an extra chromosome, while another might lack a chromosome. This is called aneuploidy, which literally means not good fold. Mutations will only pass on to children when they happen in the DNA in the egg or the sperm.
DNA contains the instructions for building your body. At the molecular level, it's shaped like a twisted ladder. Inside your cells, these DNA ladders are arranged in long strands called chromosomes. People usually have 23 pairs of chromosomes. Half of each pair comes from your mother and the other half from your father. But long before egg meets sperm, something interesting happens to your parents' DNA. Inside the cells that become eggs or sperm, chromosomes pair up. Pieces of DNA from one chromosome switch places with pieces of DNA from the other chromosome. This swapping or recombination of DNA is important. It injects diversity into the mix, and it ensures that you get just one copy of each chromosome from each parent. Each generation does its own genetic shuffle. So grandma and grandpa each mix up the DNA they inherited from your great-grandparents. Half of that recombined DNA goes to your mom or dad. Then your parents' DNA undergoes similar shuffling before it's passed on to you. To recap, you inherit about 50% of each parent's DNA and about 25% of each grandparent's DNA. But because you don't get a carbon copy of the DNA that your parents inherited from their parents, there's some wiggle room in these numbers. Depending on where the recombination happens on the chromosomes, you could inherit a lot of DNA from your grandfather and very little from your grandmother. So you and your siblings end up with different DNA mixes from your parents, unless you're identical twins. And you and your cousins get different genetic heirlooms from your grandparents. First and second cousins usually share DNA, but because of recombination, about 10% of third cousins won't share any DNA. The same goes for about half of fourth cousins. That doesn't mean you're not related. You still have common family members. And that counts as much or more than having DNA in common.